Hope
Yeah,
you
that's
don't
fine.
mind.
Yeah,
Okay.
I'll only cover public information.
Right, okay.
So let me see.
Oh, Gangadhar joined.
Let me know if you can see it.
Yeah, it has come.
Okay,
Yeah.
perfect.
Sundari, you want to introduce the speaker?
Yes, I'll do that. Yeah. Good
Yeah,
afternoon.
thank you.
Thank you for coming to today's talk. Today's talk is titled Developing a Novel Therapeutic in a Crowded MS, Multiple Sclerosis Market. The talk will be delivered by Dr. Krishnan Ramanathan. Dr. Krishnan works at Novartis GDD, Global Drug Development, as a global program head. His responsibility includes taking medicines in the late stage of research and developing them for registration as new medicines with the regulatory agencies. Currently, he is overseeing a development program in infectious disease with a new molecule targeting SARS-CoV-2. Dr. Krishnan has an academic background in biomedical engineering, and he completed his master's degree in engineering and a PhD at the Johns Hopkins University in Baltimore, USA. He specialized in the area of neuroscience and studied some of the fundamental molecular mechanisms underlying the process of hearing different frequencies of sound. Since obtaining his PhD, Dr. Krishnan has spent over 16 years working in the pharmaceuticals and biotechnology area. Originally, Dr. Krishnan hails from India, where he spent his formative years and went on to get his bachelor's in mechanical engineering at the Indian Institute of Technology in Chennai. After spending a few months working in the engineering sector, he then decided to pursue further studies in biomedical engineering in USA, where he lived for over 18 years. Over to Dr. Krishnan for the talk.
Thank you, Sundari, for the kind introduction. It's such a pleasure to talk to all of you back in IIT. The last time I visited there was for the 25th year anniversary in 2016. So I have a lot of fond memories that I still carry forward. So to do justice to the neuroscience talk, what I do is I first speak about the topic that I want to talk to you about. And then at the end, I'll do a little history of my career journey and what I've done, because it's come a long way from doing mechanical engineering in IIT to what I'm currently doing, which is drug development. So let me know if you're able to see the slides. I'll just move it on. Yeah, so I'll talk a bit about multiple sclerosis, and I recognize that all of you are engineers doing research in an engineering institution, so I'll provide as much background as I can, because this is quite heavy into treatment and diagnosis of multiple sclerosis and how we can bring a new therapeutic. It does involve a lot of data sciences, but it requires a reasonable understanding of what is the disease area that I'm talking about. So the disease area that we'll be looking at today is multiple sclerosis. It's not very common in India. At least when I was growing up, I never came across anybody with MS during my childhood there. But I did come across lots of people in the United States when I was doing my PhD and other areas as I've moved around. So it's predominantly in women. It's about 2/3 women, and you can also see from the distribution, it's quite rich in the Caucasian population. So at the further away from the equator you are, it seems like the higher chance of contacting multiple sclerosis. There is a correlation or an association with vitamin D and sunlight exposure as well, but it's not in over a couple of years. It's prolonged and genetic changes and also immune changes. With prolonged changes in vitamin D exposure. So that's the sort of the typical demographic. It's still a relatively rare disease. It's less than 1% if you look at the overall prevalence, and it varies. It comes down to very tiny fractions in the tropics. So the way one assesses multiple sclerosis is based on disability, right? If you're relatively healthy, you start off at disability score of 0, and a lot of this is done through the scoring is done through these 8 functions of the nervous system on a scale which is quite complex. But I'll just give you the kind of the high level, the engineer's version of this. You start off, if you're normal, at disability score of 0, which is a normal functioning individual. You can do all the sports activities, etc. Some level of disability, you get to the EDSS score of 2 or so. At 4, you have walking disabilities, right? You need a function of a cane. You need a walking aid at disability score 6, and eventually you get bedridden. And this happens when your nervous system pretty much is a large majority of at least the motor neurons are demyelinated. So the disease etiology, which I'll go into, is a demyelination process where the myelin sheath becomes a target of the immune system. And that is a challenge, and that leads to this progressive disability. Typical clinical trials, when we test new medicines, right, we look at the healthier side, we look at patients from a score of 0 to 6 or so, and we're doing studies in a population that's called relapsing multiple sclerosis, where you can still see them relapse, where they come present to the clinic with an acute relapse, where they have a, it's like a decompensation event, where they have problems with part of their musculature or all of their musculature, significant fatigue, unable to move, or speak or see or do some things, which resolves relatively soon, but then there's a progression that happens, which is part of that plasticity in the nervous system that you see, that resolution and the progression, which makes it very challenging to diagnose and treat as well. So clinical trials look at these healthy people relatively in the early part of this spectrum of MS. So let me move on from there. Well, another point to note as you listen to this talk on multiple sclerosis is there are things that you see that are, you see worsening or disability, you see physical symptoms, you see relapses, which are the most easy to notice, because this is an acute change of function. Because a worsening, you need to actually test yourself. You need to score yourself to know that you're going worse, because there is a part of aging that is worsening, but there is a significantly poor worsening that happens from multiple sclerosis, but sometimes goes undetected until you lose function of something. But there's a lot of things that are below the line, right? Things like cognitive worsening. You don't see it, you don't notice it, and this is classic challenge, just like what you also see, for example, in Alzheimer's disease, right? You don't see that until they become 20% poor in cognitive function, and that time it may be too late to intervene. MRI lesions, often they may go undetected, because who gets an MRI unless you have a suspected multiple sclerosis? So you can have an accumulation of lesions. You see on the right-hand side, you can have an accumulation of these dots or lesions that happen until at a point that you see a blip, which is a relapse, a clinical worsening, that you don't go and get an MRI tested. So there are many things that happen below that you don't see that lead to a delayed diagnosis and potentially a weaker escalation of treatment, if you will. And this is the challenge. This is when I said crowded marketplace, right? There has been a lot of medicines that have been approved over the last 30 years or so. So the first ones have been the interferons, right? The interferons, the way they work is they sort of tickle the immune system to act on itself. And there's a lot of history to MS, which there's an assumption that it sort of correlates post-infection of, for example, Epstein-Barr virus. So the immune system in some ways, and these interferons also have antiviral properties, by suppressing the response to the virus, you are then somehow helping MS. These interferons have done relatively well in suppressing new lesions, but they have not done that well in stopping the progression or the clinical relapses. And then that led to the discovery of multiple other compounds. And until natalizumab in 2004, which is a monoclonal antibody that prevents egress of immune cells into the CNS. So it basically thickens the blood-brain barrier and stops these infiltrating immune cells to go in, which is a profoundly good treatment, right? You could show that you can reduce the damage to the CNS from your own body's immune system, but it led to a bunch of other things. So there are some brain-resident viruses, like the JC virus, and this leads to complications where this virus then starts developing in the brain and leads to an issue that you never thought you would have, which is progressive multifocal leukoencephalopathy, or PML it's called. And people are worried about PML if you have MS, because what you do, most of these treatments suppress the immune system, that then lets the brain-resident viruses, which are otherwise dormant, to take over and destroy other parts of the brain that are not needed. That's a sort of a damage, shall we say, collateral damage that happens. Natalizumab was approved and then withdrawn and then reapproved to help patients because the patients wanted it. They said, we'll deal with the PML, but we can't deal with multiple sclerosis. So that led to a slowdown of then future antibodies that were developed until alemtuzumab came in, which was a repurposed antibody. It was originally indicated for cancer. Most recently, ocrelizumab, which is an antibody to CD20, which is very similar to the molecule that we developed in Novartis, and I'll explain that in just a bit. So you see this checkerboard of medicines And if you put them out in a landscape of high efficacy versus good on safety, right, you get one or the other. You get a very potent, efficacious medicine, but it somehow conquers the immune system in a way that you have then other issues. You have, infections popping up, and you don't want that in multiple sclerosis patients. So the way to understand this is to sort of go back a bit to the biology of the immune system. And I'm not the immunologist nor the expert here, but if I just go to how our immune system builds tolerance, right? Usually you don't attack things in the body. The immune system is tolerant to things in the body. You are attacking pathogens that are foreign. So a part of the immune system, which is the adaptive immune system, is responsible for developing this tolerance and sometimes the breakdown of this tolerance as well. As in other diseases like rheumatoid arthritis, psoriasis, multiple sclerosis, this breakdown of tolerance happens maybe after an infection from Epstein-Barr virus or something, but this breakdown leads to these B cells somehow becoming then pathogenic. Certain B cells become pathogenic and they start presenting antigens that are in the myelin of the nervous system. And then that activates then the T cells, which are either the CD8 positive or the cytotoxic cells that go and attack these myelin. So the B cells are sort of at the higher end of that cascade where this breakdown of autoimmunity happens. And there was a view that if you then are suppressing these B cells from presenting these antigens, you might actually take back the disease, if you will. It may lead to other complications, right? Because B cells are also important in host defense, in defending against foreign pathogens. But you rarely know of a person, at least in today's world, with great antibiotics, great antivirals, and all of these, unless you have like a pandemic going on like today, right? I mean, I just correct myself there. You don't think of middle-aged people dying of infections today. It happened 50 years ago, but it's not happening now. So one could think that by suppressing these B cells, you can turn down that immune process by which the tolerance is broken. And one way to do that is to look at some receptor on the B cells and then put an antibody towards it that then takes out those B cells. These antibodies have a mechanism of killing by cytotoxic ability or ability to attract other cells that are cytotoxic. So one receptor that's on these B cells is a CD20 receptor. And you see it's constitutively expressed in this lineage of B cells, not in the stem cells, not in the pro-B, but in the place of maturation where these memory cells are being activated. And that is the area where this pathogenesis develops, where your breakdown of tolerance happens. But then this also CD20 receptors are not present in late plasmablasts or plasma cells, which are important for antibody generation, and examples like SARS-CoV-2 antibodies. So one could think that if you don't take away all the B cells, if you are stopping some of those in the middle that may be responsible in the pathogenesis, then you still allow some B cells to develop and produce antibodies, etc., in the long run, but then you stop the pathogenicity, you stop the disease. And lo and behold, this was good. There are many B cell-directed antibodies, CD20, and what is different to what we developed was ofatumumab. Actually, we licensed it. I didn't discover it, but we developed it. It's a fully human antibody, so it's relatively silent to the immune system in of itself. The other antibodies that were on the market before rituximab are chimeric antibodies. So they have some immune sequences that are from murine. And this leads to the antibody itself being immunogenic. So our immune system then reacts to this. And so you need to give a pre-treatment with steroids to stop the immune system from attacking the antibody itself. And the other advantage is that this binds to two epitopes of the CD20 receptor that gives it a better affinity and avidity. And it's also closer to the cell surface. So it has a slightly better killing process. And essentially what that means is smaller doses of this antibody could do better in the treatment pattern. Because of these properties, one could think of giving this antibody subcutaneously. So you don't have to go intravenously. Most antibodies are given intravenous, but if you are fully human and you're not immunogenic, because going subcutaneously, you cross these dendritic cells, which also react to foreign antigens. And if you have a foreign antibody, for example, with murine sequences, you'll then develop an immune reaction locally, and stop the antibody right there. So because this is fully human, you could go subcutaneously, and going subcutaneously gives you better access to the lymphatic system, which biologically in the lymph is where these activation of B cells and T cells take place. So we thought that the antibody then becomes better targeted biologically to those cells that are pathogenic. So there are B cells in other parts of the tissue that are not doing this pathogenic function, right? They're not activating T cells, which happens in the lymph node. So we thought that this design was very elegant, and hence the development process that led to, eventual licensure of this molecule. I go through very quickly about this clinical data, and it's at a high level. Each of these charts can be explained for many hours, but I'll try to do it in a way that it's helpful for our discussion, but not going into the pharmacological details of this. So we actually didn't do this, but another company, GlaxoSmithKline, did the phase 2 study, and a phase 2 is a dose-finding study, and then we did the phase 3 study, and we used this to model the dose that we thought we could take forward. So in the phase 2 study, you're looking at number of T1 enhancing or gadolinium enhancing T1 lesions, and what are they is gadolinium is this heavy molecule that penetrates the brain only when the blood-brain barrier is open. Under normal blood-brain barrier conditions, you don't see gadolinium penetrating. So that means that either T cells have gone into the brain to attack the neurons or the myelin, and that has carried some of these gadolinium with it. So when you have these enhancing lesions, you have active disease. You have active immunological presence of these inflammatory cells in the brain. And so you see here with placebo patients, there is a continuous new lesions that is taking place, but with patients treated with any of these doses, 3, 30, 60, either given every 12 weeks or every 4 weeks, you see a suppression of these new lesions. And we needed to come up with a dosing mechanism based on this. Another way to look at this is just mean number of new lesions, right? Are there any new lesions, not the cumulative ones? Are there any new lesions? And you see that even on patients who were treated with placebo were switched, because ethically you don't want to give them no treatment for a long time. So they were switched to the 3 milligram dose, the lowest dose in this study. But you see a good effect as low as 3 milligrams, which was surprising because previously used these molecules or other approvals of rituximab and others were used over 600 milligram, which is quite a substantially high dose of this antibody. So we were surprised to see this in the phase 2 data. And when you model this, you see what's happening to the B cell count. And this is circulating B cells in blood. You see that with a single dose of 60 milligrams, these red lines, you pretty much take out all the B cells that are circulating. So giving subcutaneously this molecule was more potent than giving it intravenously, because the B cells predominantly, they're sitting in the lymphatic system and you could target it. So we were really surprised by this activity at 60 milligram. And then you see even at 3 milligram, you see a dose response here with the B cells going down very quickly. So we did a modeling, a physiological model on what would be the dose and what is the target you want to get to. And we used this correlating with the lesion data that a target we want to get to is below 8 cells per microliter. That's the dotted blue line. And we wanted to get the circulating cells below 8 cells per microliter. And we thought that we could do that with a dosing regimen, of 20 milligram monthly, right? Because you stop the replenishment between those doses, because each dose is active for a few weeks based on its half-life. And while you have new production of B cells happening, you want to keep them at a low level. And we had to get to this level quickly, so we wanted to give the first few doses more quickly. So giving it weekly for the first 3 doses and then monthly was sufficient. And that was the regimen that we took into phase 3, fingers crossed, hoping to have a successful outcome. So we did a number of other studies in addition to the phase 3, and we usually give these studies a name, so it's easier to talk about it. Otherwise, it's a long title, clinical comparison of X versus Y, randomized, double-blind, placebo-controlled, and all of those things. So Asclepios 1 and 2 were the name of the 2 studies that were phase 3 studies. Then we did some parallel studies. One was to approve a new presentation, not just a prefilled syringe, but an auto-injector where patients don't see the needle. You can just press it to your skin, either your thigh or wherever you want to inject yourself, and the needle comes down by itself and retracts. So it's a spring-loaded device. So we did this testing in the study called Apleos. Then we had to do a separate study for Japan, which was called Apolitos. And these names were all created by us to help with the branding of anything that ends in OS was ofatumumab subcutaneous somehow, to instill an ability to have this dialogue with the stakeholders in the wider group. So the phase 3 study, just the design, I could speak hours for on this. We compared ofatumumab with a oral comparator, teriflunomide, and teriflunomide has also antiviral properties and anti-inflammatory properties. It does something to the DNA intercalation, which in terms of rapidly dividing immune cells, it stops them from doing that. So it has a reasonably good effect on immunosuppressive mechanisms. It is also a brain penetrant molecule, so it could do something in the brain as well, which we don't know, but it was approved for multiple sclerosis treatment, and it was used as a first line because its efficacy was not great, but its general tolerability and safety was good. Unlike these interferons, which had to be injected very often, every 3 to 4 days, or sometimes every week, depending on the molecule that you pick. So we picked a comparator that was relatively, shall we say, easy to deliver in a phase 3 study. You can have oral medication, and you could do a double-blind study. You can give blank tablets and a blank injection, although instead of having 2 injections, which would make it a very significant injection burden for patients. And these placebo-controlled studies are important, and they're ethically permitted, or not placebo-controlled, comparator-controlled with double-blind over a certain period. And so we could do this because teriflunomide itself was an approved medicine for these patients with relapsing multiple sclerosis. So we did a lot of other things from a data science perspective to make this a flexible design, which is an event-driven study where you look at clinical relapses, look at progression events, so that we don't have to expose them to this design of double-blind for more time than needed. Right? It's not a fixed duration study, but it's a flexible duration for just the amount of time for which you need enough delta in the design between these 2 treatments. We looked at a number of endpoints. The primary endpoint is really preventing clinical relapses or reduction in annualized relapse rate. Other endpoints, which are really important, is preventing the worsening. And these worsening events are rare because if you remember the EDSS scale going from 1 to 6, you have to worsen 1 point or 1.5 points on that scale, and you have to stay worse for 3 months or 6 months. So you don't want too many people going worse on your clinical studies because it is still a double-blind study. So you try to power your study to a point where if you see a difference, then you can stop it. And on the right-hand side, we looked at all these standard biomarkers, which are imaging biomarkers, T1 enhanced gadolinium enhancing lesions, T2 lesions, which looks at cumulative damage to the brain, and an interesting molecule, which is another biomarker, which is called neurofilaments, which is a protein that's part of the cytoskeleton of neurons. So you could see higher levels of neurofilaments in blood if there's damage to neurons in the brain. And there was an assay that was available while we were doing the study, so we amended the protocol to incorporate this as an endpoint, and it was included as part of the design testing criteria in the hierarchy of testing. And finally, we also look at brain volume loss, which is very interesting as I will go into the data. So I'll go through the summary of these results in a few minutes, and then we'll open up for Q&A. So needless to say, we had good data a year and a half ago to show that you could reduce the relapse frequency significantly. So the relapse frequency that you see eventually obtained was 0.1 or 0.1, which is 1 relapse every 10 patient years, which is quite low. MS patients relapse quite often, often more than once a year. So getting to this relapse frequency was a significant achievement already for these patients. Well, this is the beauty of the results that we saw. We saw nearly no new lesions, if you look at it. So the number of new lesions seen were about 0.01 or 0.03, which on average was 1 lesion in 50 scans that we did, 50 MRI scans, which were done annually, right? And these patients develop usually more than a lesion. They started off with 1.3 lesions on average. So getting to this point, even comparing it to another active drug, teriflunomide, which got the lesion frequency to 0.4, which was 1 in every 2 lesions or so, 1 in every 2 scans, was a significant achievement. So there was a very good suppression of the formation of new lesions. So new disease will get stopped. However, lesions that have already happened will continue to do damage to the brain because we're not stopping that. We're not stopping the neurodegenerative process. Another beautiful result is looking at neurofilament levels, and you see, So even healthy people have some level of baseline neurofilament in blood, right? And there is a remodeling process that goes on. And typically we think that this is low. The mean concentration in picograms per ml for healthy is probably between 4 and 5 or so. So we got pretty close to the healthies. We don't know the healthy levels yet. There's still large-scale studies that have not taken place. We use this as a method to see what is the difference between ofatumumab and teriflunomide, and a significant difference. So you're protecting the neurons from dying in this. And then in contrast, we looked at overall brain volume loss in imaging processes, just looking at MRI scans and integrating them geometrically to look at three-dimensional volume. And here we didn't find a difference. So this was extremely surprising for us. So there is something that teriflunomide is doing that is either protecting the brain volume from going down, or there's something that ofatumumab was doing that you don't see. And interestingly, we could see a change in the slopes in the second year, but if you index them all to the first year, the brain volume loss with teriflunomide was not very high compared to the brain volume loss with ofatumumab, which was extremely shocking for us. One of the beliefs here is that there is a lot of infiltrated cells. So these images are not able to recognize between, infrastructure or cellular structures there, neurons, astrocytes, glia, microglia, all of those, versus inflammatory cells that are coming in. And what we are seeing is a sort of a pseudo attrition of the brain, we think. And we're doing a lot more analysis on this in collaboration with the Oxford MRI Institute to see if we can disentangle this in a biomarker basis. Extremely challenging, and I wanted to put this out to you since you're all in the space of engineering, to look at these scans, to also think about these processes and how we analyze it. I'm not the expert on this, but certainly there's a large team that I'm working with who have looked into this in detail. Any clinical study will also have to look at the safety. So we look at this from just a reporting of adverse events, and from an overall gross number basis, any adverse event, the frequency of report of adverse events was no different to teriflunomide. There were some signature differences. Injection reactions were higher in ofatumumab by 5%, and this is what we would typically expect with a therapy that depletes B cells, right? And I'll go to that in another slide. And then you see relatively balanced. There's some more signature differences which are very pharmacologically distinct between these two molecules, but I won't go into the detail. But it's important to note that the safety didn't cause any additional concern in ofatumumab, and also from a severe adverse event perspective. And these are very clinically relevant: new infections, malignancies, things that we make sure that statistically there is no difference, that somehow stopping these B cells didn't unleash other activities in there. And now on the injection reactions, you see these blue bars are injection reactions from ofatumumab, and the gray bars are from teriflunomide. And these are injection numbers: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc. And the difference is only with the first injection. So you see a reaction whether you give placebo. So these people on teriflunomide got placebo injections. So there is a reaction to the first injection. But there is a difference versus ofatumumab. And then that difference sort of disappears statistically over the newer injections later on. And what's happening here is you start off with a high level of B cells, and the first injection depletes some of these B cells. And by the time you come to the second injection, you're already at low levels of B cells. So the reactions are not skin reactions. These are systemic reactions. So when B cells die, they have pockets of cytokines in them, and you see these cytokines released. And this is like typically when you have an immune reaction, etc., when you have a fever, and you are seeing a cytokine reaction. And usually you give steroids to treat that, pre-treat them with steroids before the injection. But we did this in a blinded study, and we showed that you actually didn't need these steroids, that these reactions were quite manageable. And even giving them steroids, and inside this is a blended data of with and without steroids, caused a bigger reaction with just teriflunomide or placebo injection. So if you look at that delta and the relative intensity of these reactions, one didn't need to give steroids, which themselves caused reactions. And so that was the advantage of going subcutaneous. So we think that given this package of good safety, good tolerability, and extremely good efficacy, that this would help change the treatment landscape. Time will tell over the next 10 years how the practice changes. And in summary, we could show a significant reduction in relapses, extremely good data on MRI, lower levels of NFL, which was the first time anybody had done a measurement on this marker, and we were able to do it, and a favorable safety profile. So all in all, the study was presented in 2019 at the ECTRIMS meeting in Stockholm, which led to eventual approval in August this year with the FDA. I wanted to reiterate and the importance of why we do this in drug development. So this molecule was originally invented by a small company called Genmab, and they worked on it for 6 years. And at that time, they were thinking to develop this for cancer indications. And what are the types of cancers when you have, for example, lymphomas? And what happens in a lymphoma is just the B cells start proliferating. You have 3 orders of magnitude more B cells than you typically would have, and you have a cancer. And it takes away resources from other parts of the body, other stem cells from proliferating, and so you lead to significant downstream damage. And you can stop that by giving this antibody, and there's approvals of rituximab there. But it's not well differentiated versus rituximab, right? Because cancer therapies, combination, and these toxicities, the safety profile of this, the dosing, were not advantageous there. So rituximab was still the standard of care. They couldn't break that in. They handed it over to GlaxoSmithKline, who worked on it for a number of years, looked at multiple other indications, arthritis, pemphigus vulgaris, multiple sclerosis. They did all these phase 2 studies. And then we purchased it from GlaxoSmithKline in 2014 and 2015, 2 different agreements. One was for the oncology part, the cancer part, and 2015 was for the autoimmune and multiple sclerosis part. And we thought that the only place where we see a big new benefit, even though it's a crowded market, is in multiple sclerosis, because there you have this very potent reduction of new lesions through B cell targeting therapies, and at the same time, a significant need of therapies that have this balance of being able to do it without causing damage and other things like brain resident viruses popping up, infections, and all of those. So it took a long time. We licensed it, and even after bringing it in 2015, I joined the team in 17. I worked on this for 3 years. We had a plan to spend over $300 million for the phase 3 development, and that's the cost it takes to approve a new medicine. The study was done in 38 countries. We had over 370 sites, each site with 2 investigators. So when you have these meetings, these are very large meetings over webcast or sometimes in person to train the investigators on this. So a lot of this was done by us over the last many years that led to the eventual approval. So I've been working on it for a while. So before I close, I wanted to give you a flavor of what I've been doing and how I ended up here. And so I was born in Delhi very long back. I'm Tamilian, but born there, and I speak Hindi quite fluently since I grew up a bit in North India. Spent a number of years in a few other cities, Nagpur, Pune, and all, and then eventually ended up doing my studies in IIT, which I had great fun in Chennai, or Madras at that time. I went on, I worked for a year in Mumbai, which was here in Larsen and Toubro, and I was a mechanical engineer. I was doing design engineering. And during that time, something happened, and I said, you know, I want to explore a different subject. And I applied to biomedical engineering, and I got into Johns Hopkins. I went off to Baltimore, spent the next 6 years there, master's and a PhD. My first job was in Chicago. I was in a consulting organization with McKinsey for 3 years. So it helped me get a flavor of what happens in management and decision-making, all of it. I came to Philadelphia to work with GlaxoSmithKline, and this was a role in R&D finance. So since I had done some consulting work and I had the language to talk to the people in R&D, I was helping management make decisions on which diseases to invest in, which not, over the portfolio of the medicines in GSK. I did that for 3 years. Then I went to a small company in San Francisco for 2 years. I somehow thought I'd marry this passion of this understanding financial analysis and developing medicines to be entrepreneurial. That company wasn't successful in 2 years. Our medicine failed in phase 3. Then I joined Novartis in San Francisco. I was then moved to Boston or Cambridge, worked there for a few years on vaccines portfolio. Then I moved to Basel in Switzerland, which is where I am now. And then I started changing again, going more into classic drug development, less from financial analysis or asset development to more classic understanding, learning, clinical trial process, pharmacology, all of these things. I spent 3 years in Moscow as an assignment because Russia was our second largest clinical facility. First was United States, where we had over 1,000 sites, and in Russia we had over 450 sites. Meaning these are institutions, right? Even if it's a small clinic, it's an institution or a hospital. And I visited a number of these to ensure that we had good quality, good training of the investigators. So very large clinical team, but a great experience, challenging experience as well from a language perspective. And then I'm back to Basel now. So that's where I am. I'll stop here, and I would be really pleased if you all turn your camera on and ask me questions and whatever things I can help you with. I hope this was useful and not completely off into a phase of pharmacology development.
Thanks a lot, Krish. I really enjoyed that talk. I'll request my other friends here to ask questions. I have a couple of questions, but I think I'll go at the end.
Sure.
Anyway, Gangadhar, I think you can meet Krish right now. Are you there?
Hey, Gangadhar, how are you?
Hi, Krish. It was very interesting talk. So I work in Lausanne with MindMaze. Perhaps you heard about it. So
I did.
Yes, I'm responsible for clinical development at MindMaze, looking after a number of clinical trials. Definitely not at the scale of yours, smaller in scale, but in the digital therapeutics domain. It was very interesting to know the other side of the pharmacological development. At the moment, I don't have much questions.
No problems, no problems. Nice to meet you, Gangadhar. Happy to catch up later on.
Right.
We're in the same time zone, I imagine. Yes.
Right.
Anybody else? Anybody has any questions? Okay. Hi, Gangadhar. Krish, I have a couple of short questions. So slightly.
Sure.
So you were talking about how expensive the whole process of drug development is, right? So now some companies are looking at building these multi-scale models to understand the drug action from molecular level all the way to the symptoms. So there's a whole field called computational neuropharmacology. I mean, that's a buzzword that people are. And it's a lot cheaper to develop the simulators. And then if they work, then you can move on to animal studies and clinical trials and all that. Now, is Novartis into that kind of activity, or are you still looking at more classical drug development?
I think we would not shy away from it. Are we into it? It's hard to tell. I think the success in the future will determine. So we are looking at a lot of in vitro studies which can somehow discharge risk in later development, right? I think phase 3 failures are the most expensive. Phase 2 failures then, and then phase 1. So we typically try to take compounds into the clinic because a lot of these times, including in multiple sclerosis, animal models are not very informative. So the way we test new drugs in MS in animal models is using artificially induced lesions in mouse brain, and this is done through injecting a virus like equine encephalitis or something that you put in, and you generate lesions, and then you treat them with different modules. And all of these have initially shown that MS was a T-cell derived disease, that you need to stop the access of these inflammatory cells to the CNS, and somehow you can stop MS. And so you see a lot of these early therapies were T-cell dependent until somebody tested rituximab in MS patients and saw really the lesions went away. And they said, well, it's a B-cell disease, but these animals don't have the same biology of B-cell to T-cell interaction. So I think those experiences still leave some gaps between good translation of early development to later. But if there's mechanistically, if you can incorporate some of these, right, into showing that this biology is well replicated in these microcultures or however we want to test it, then I think it carries a much better weight to taking it to later development. Eventually, any of these will have to go through a large phase 3 because of safety reasons, right? We had to do this partly because of efficacy, but none of the regulators would allow us if we had less than 2,000 patients in our clinical trials. So we had to get a total of 2,000 patient database over this period. And so that then becomes, you have to invest in the late stage, but you might be more sure of taking compounds to late stage. Because we have still, I think as an industry, the failure rate in phase 3 is close to 50%. And that's very expensive. When you invest $300 million and you swing a coin toss and says, half the time it's negative, it's very expensive.
So a second question is, I think there's some evidence that there is a glucose metabolism impairment, right, as a basis of multiple sclerosis. We found something similar in case of Parkinson's and Alzheimer's in our modeling effort and also general survey for Alzheimer's and modeling for Parkinson's. We found that that's the kind of thing that seems to explain a lot of phenomena. Now, is any of your drugs working on that basis to fix the metabolic impairment?
Yeah, none of these. There is some evidence that teriflunomide, our competitor, has some benefit on mitochondrial function and also in changing the electrodynamics of the cells, which might lead to better metabolization of glucose in the brain. Now, the typical diseases like MS and others that have an initial inflammatory attack and then a later on neurodegenerative process, most of the drugs are looking at this inflammation process where you can stop new immune cells from coming in, like ofatumumab or teriflunomide and others. There are very few that look at the neurodegeneration, and this glucose challenge that you're showing in these cells that don't metabolize glucose well comes when these cells become hypoxic or they are in the neurodegenerative phase. And there are very few medicines that have shown effect there. And that leads to these challenges in developing new medicines for Parkinson's, Alzheimer's, etc. And also progressive multiple sclerosis, where you've stopped relapsing, but you continue to progress, which is seen as post-inflammation neurodegeneration.
What about combination of brain stimulation and drugs? Has it been tried for MS? Because that kind of thing has been tried for Parkinson's. Because that
in Parkinson's, it is more possible because you know the exact area where there is damage, where you know that the dopaminergic neurons, where they reside in the midbrain, and you know how to stimulate them or how to change the modulation. For MS, the lesions can happen anywhere. They can happen in the pons, cerebellum, anywhere. So it's hard to do that.
Okay, thank you. Hey, anybody else? So looks like there are no more questions. I think we can thank the speaker once again. And please let us catch up again. And I want to discuss a lot of
that.
And
super.
Ganga, since you're right there, you guys can chat up anytime.
We'll do that. Yeah. Thanks, Srini. Thanks for the opportunity. Thank you, Sundari, for the kind introduction. Yeah.
Thanks a lot.
Bye.
Bye.
Take care. See you.